mean Scary Movie. Wait, you guys haven't watched Scary Movie, huh?
I watched all.
I forgot. I like in Scary Movie 2, and it's like tucked in or tucked out, and it's like that. Oh, it wraps around. I love Scary Movie 2. I want to watch it now.
No, you don't.
Yes, I do. I've seen crazy shit. I don't care. Nathaniel gets your dad. What is he doing? No, I know. Yeah, I've seen my share of group chat videos. Like, do you know the one where it's like the bike and it switches to the anal prolapse? Yeah, that was crazy. Okay, anal prolapse.
Let's go ahead and
They're gonna get me.
It is 5 after, so people are still on their way. They'll get caught up, I would imagine. So this morning in lecture, we had sort of ended on this slide. We've been talking a little bit about the basic components of blood, kind of some basic measurements that we can take. We also looked at red blood cells in a little bit more depth. Of course, we'll return to those when we talk about the respiratory system in our next unit. And we had started to talk about white blood cells. Really ended pretty well, because what we want to do today is we want to start by learning to identify these white blood cells under the microscope. So that's going to be kind of one of our primary focus today is to look at these blood smears and learn to identify the different blood cells that we come across. Just one of those things that takes a little bit of practice. So real quick, let me refer you back. I showed this slide this morning. I want to refer
I
you
forgot
guys
what
back
Rhino
to
looks
this
like.
slide right
It's just
here. This
when
is a pretty
photos
good
get
roadmap
burned.
for the things that we find in the blood. I also have listed some really basic functions of the different white blood cells. So if we are asking about function, really I don't expect you to go into a huge amount of detail other than what's listed like kind of right here and kind of what we'll talk about in just the next couple of minutes. So this is a pretty good place to start for what I'm looking for as far as the functions of these. Let's go ahead and take a look at our white blood cells and take a look at what their jobs are. So again, your white blood cells or your leukocytes number somewhere between 4 and 11,000 for that box that you have on your desk, right? Remember I asked you guys about the box this morning, the little cubic millimeter of blood that you've got, right? So inside of that blood sample that you've got somewhere between 4 and 11,000 white blood cells. Our white blood cell numbers are going to vary fairly considerably, depending on what potential pathogens you might be fighting, right? What might be going on inside of your body. What we're going to see over the next few minutes is that some of these will spike in response to some things, and then other things will spike in response to other things. So with a routine blood draw, what we can do is we can do what's called a differential white blood cell count. And you're actually going to do one of these today, and we can see if any of these are abnormally colored, right? And that will give us an idea, along with other tests that we could run, of what might be going on with the patient. So with that in mind, let's go ahead and take a look at these. So as we mentioned this morning, our white blood cells are divided into two groups based on whether or not granules exist in the cytoplasm. So the ones that do have granules in the cytoplasm are called granulocytes. So let's go ahead and take a look at a couple of examples here of some granulocytes. our first one are neutrophils. Okay? Neutrophils are something that you're probably going to be very tired of looking at because they're very plentiful in our blood. They account for somewhere between 40 to 70% of all of our white blood cells. So they're very plentiful. Like I said, we're definitely going to get a good look at these guys today. And like all white blood cells, neutrophils have a, I don't have a purple pen. If I had a purple pen, it would work a little bit better. But neutrophils are these cells. They're a little bit bigger than red blood cells. But what really characterizes them is the fact that the nucleus is very distinct. Okay? So we can have a nucleus here, and this nucleus can have multiple lobes to it, so that it kind of looks like this, right? So this one right here, for instance, I have 1, 2, 3 distinct lobes here. All right. The one that's on the board, there's also 3 distinct lobes. Sometimes it looks like there might be more than one nucleus because the little string that connects the 2 lobes sometimes is kind of hard to see. Okay. And then the other thing about neutrophils is even though they are granulocytes, the granules are actually going to be kind of hard to see under the microscope. They're just really, really tiny. We're not zooming in quite enough to see them. But the cytoplasm does have little tiny granules. Sometimes that nucleus can be, like those lobes can be kind of stacked on top of each other, right? So it can look kind of like this, right? Where you've got multiple lobes that are kind of stacked on top of each other, kind of like that. Sometimes you get a nucleus that looks like this, where you've got your nucleus like that, and then there's the other lobe that's kind of down here. They can really vary quite a bit in terms of the way that they look. But this is a good example. Some of these guys can have as many as 7 lobes. So, and I'm sorry, that's not showing up too far. But some of these guys can have as many as 7 different lobes. So they can have quite a few different lobes attached. So neutrophils, like I said, you're going to get really tired of these guys by the end of the day. We're going to see hundreds of them today. They're active phagocytes, so they kind of look around for things to destroy. They can phagocytize red blood cells as they die. And of course, during a viral infection or a bacterial infection, we would see their numbers going up considerably. Okay, so they're a great example of just a phagocytic white blood cell. The next one that we have are eosinophils. Okay, eosinophils are also a granulocyte that has a lobed nucleus, but that nucleus is almost always bilobed, meaning that there's only going to be 2 lobes to it, and they tend to be a lot easier to see. Let me see if I can get this going here. I might have to do this in black because I don't have a good red pen. I apologize for that. Your cell is right here, and then you've got this very distinct sort of bilobed nucleus that kind of looks something like that. So it's pretty distinct. We're definitely going to see eosinophils today. The other thing that, of course, sets them apart is that the granules are way bigger. So you're going to see lots of bigger granules in your eosinophils than you did in our neutrophils. Like I said, in your neutrophils, it's going to be hard even to see the granules because they're so small. But in our eosinophils, we're definitely going to see granules. Eosinophils get their name. This might help you remember this. Eosinophils get their name. Eosinophils get their name because they take up a stain called eosin really easy. They're eosin phials, right? A phial is a lover of something, so that's the suffix "phil" comes from phile, a lover of something, right? eosin is this stain. Sometimes it's called eosin red, but it does have a very dark red color to it. If you make blood smear slides, you're going to get eosin all over your fingers and your clothes and stuff like that. Fingers will be red for weeks if you get it on your skin. But these eosinophils take up that red stain very easily, and as a result, they appear pretty red, especially the granules appear really red under the microscope. So we'll know when we see an eosinophil under the microscope because they're dark red granules, and they're pretty easy to find. So even though their percentage, they only account for somewhere between 1 to 4% of all of our white blood cells. Even though that percentage is pretty low, we will see them today. We'll see them on the first slide that we look at, which is a normal blood smear. But then we also have a slide for you guys of a patient who has eosinophilia. Okay? Eosinophilia, we add on the IA, eosinophilia is any condition that results in elevated eosinophils. Okay? Eosinophils tend to be elevated most commonly when we have parasitic infections. So, do we know what the most common parasitic infection in the world is? Ringworm? Not ringworm. That's actually a fungus. That's not even a parasite. That's just a fungus. It's just called ringworm. That's a good guess. Malaria is definitely probably the one that's going to be the most prevalent, right? Especially at equatorial latitudes. When you get closer to the equator, malaria is a big concern. It's a blood parasite. It's transmitted by mosquitoes. We don't obviously have to worry about it here in Southern California. Yeah, who knows what the next 10 years of climate change is going to bring. As of right now, we don't have to worry about it. But when we have malaria, we have elevated eosinophils. That person now has eosinophils. Okay? So we're going to see that slide when we get around to looking at it later on this afternoon. It's just Chucky Jam full of eosinophils. So we're definitely going to see it. But you'll see them on the normal slide as well. Okay, so we have neutrophils that we'll see. We have eosinophils that we'll see. Our third, and eosinophils also can be elevated during allergy attacks, but they tend to be we tend to normally associate them with parasites. Our third and final granulocyte that we have are basophils. So this basophil looks really, really similar to the eosinophil. It has a nucleus that can be U-shaped or bilobed. In some cases, it's kind of S-shaped, but in almost every case, it's almost also impossible to see the nucleus of these guys because basophils have big granules that stain really dark purple when we make blood slides. They take up a different stain than eosinophils do, and as a result of that, they stain a different color. But they're really dark when we see them. And you can also see here the basophils, you've got your box of blood on your desk that's a cubic millimeter, right? Basophils, there's only going to be between 20 and 50 basophils in that box. So this is like the great white buffalo of your white blood cells. There's just not very many of them. If we find one, that's pretty awesome. We saw 2 basophils this morning, and another one that was questionable. In my lab yesterday, we only saw 1 basophil, and I haven't found any this semester yet. So I'm relying on you guys to help me find basophils. But we might find one today. It'd be really cool if we do. But they're really, really rare to find in the blood. One really cool thing about them, though, is that even though they aren't super common in the blood, is they play a really big role in the inflammation response. The granules of basophils contain histamine, and I think we all probably know histamine or have heard of it anyway, right? Histamine is a really powerful vasodilator. So when we have damaged tissue, whether it's from like actual tissue injury or whether it's a bacterial infection or something like that, whatever it is, that area is going to give off, those cells in that area are going to give off chemicals that are going to call basophils into the area. And basophils are going to release histamine, and it causes the blood vessels to enlarge in that area, to vasodilate. And we bring more blood into the area and consequently more white blood cells into the area, right? So these guys are really instrumental in the inflammation response because they do release histamine, right? We know histamine as well because when we're having an immune response to either a perceived or an actual pathogen, sometimes we get the blood vessels in our sinuses dilate, and we have our sinuses filling with fluid. That gives us a stuffy nose. And then simultaneously, while histamine is a vasodilator, it's also a bronchoconstrictor, meaning that it constricts your airways. So what it does is it brings fluid into your sinuses, and it also makes it a little harder to breathe, right? So what do we often take to help us alleviate those symptoms when we've got allergies or a cold or something like that? We take an antihistamine, right? We take something to block histamine from binding to its binding sites to help us to relieve that sinus pressure and stuff like that. So something like Benadryl or Sudafed are great examples of antihistamines that can block the histamine released by basophils because it's a really powerful vasodilator. So kind of an interesting thing. They're not very plentiful, but they have a really powerful effect on our inflammation response. Okay, so we'll definitely see neutrophils. We'll definitely see eosinophils. Basophils, we'll see. We'll see what we can find. All right. And then agranulocytes. We have two types of agranulocytes. Again, lymphocytes are going to be pretty easy to find. Lymphocytes are usually characterized like they're shown right here, where there's a really large round nucleus, and then you'll notice that there's just a sliver of cytoplasm that's visible. The nucleus occupies most of the cell in lymphocytes. So they're pretty easy to see, they're pretty easy to identify, there's nothing else that really quite looks like them. When they're immature, there's a little bit more cytoplasm visible, and then by the time they mature, they look like we see right here. Lymphocytes play a role in our specific immune responses. These are the ones that become B lymphocytes or B cells and T cells, right? So depending on where they mature, they play a really specific role in our immune system. And they're very, very active for lots of different things. So they're pretty important little cells. Obviously, we're not going to be able to tell the difference between T and B lymphocytes under the microscope today, but we'll definitely see these guys. And then finally, our last type of agranulocyte are monocytes. Monocytes are generally characterized by their size. These guys are blue, and we'll be able to see several of them under the scope today. If we have one that's nice and pretty and round, like the one that we see here, there's a C-shaped nucleus that you can see. But again, the size is kind of going to be the giveaway there. Well, like I said, we'll put some slides up on the scope here in just a minute to take a look at them. But you'll know when you see them. Monocytes are also phagocytic. When they're in non-blood tissues, they become macrophages. So we've heard that term macrophage before, especially if you've talked about the immune system in other classes. Macrophages are monocytes that have just left the blood. When they're in the blood, we call them monocytes. When they're in non-blood tissues, we call them macrophages. Of course, these guys also, because they are so strongly phagocytic, we see them in long-term infections. We see them even in viral infections, like short-term viral infections. And then, of course, we have a disease named after monocytes. What's that? We probably all have heard of it. Mononucleosis, right? Mono, right? You get mono, the kissing disease. That's transferred via saliva, and that's one way to get mononucleosis. Right? But that one's named after monocytes. So we see that it's the levels of monocytes are elevated during different periods. Right? So the idea here is that all of these guys have slightly different jobs. Right? If you're having a really strong inflammation response, we're going to see that your basophils are elevated. If you have mononucleosis, we're going to see that monocytes are elevated. If you have malaria, a parasitic infection, we'll see that eosinophils are elevated, right? So doing one of these differential blood cell counts like we're going to do today is a really helpful thing because we can compare your white blood cells or your patient's white blood cells to kind of the normal values. And if we see that one of those is abnormally elevated, we can explore a little bit further. Okay. So I'm going to put up on the board a slide, and let's take a look at what we're going to spend some time doing today. So we have 2 slides today in the back of the room. We have a normal blood smear, and then we have an eosinophilia. In just a couple of minutes, I'm going to ask you guys to go get slides. I would like us to all start with the normal blood smear, and then we'll look at the eosinophilia second. So make sure that you take an extra good look at the label. And then this is what we should see. It's focused here. Look. So, our blood smears, and I'll help you guys get good pictures of these so that you can have them for your study. So on low power, a blood smear doesn't show us very much. This is what we see on low power. And you're literally going to see millions of mostly red blood cells. Okay? So there's no real reason to spend time at low power. I'm going to zoom in a little bit. Again, more blood cells. I have one extra objective lens than you guys do. So this is my 20x objective. And you can see here that with the exception of a couple of platelets, this is all red blood cells that we're looking at right here. I'm going to go to my high power that you guys also have and just kind of take a look here. So this is my high power objective. And we can see that, again, we're looking at mostly red blood cells here. Remember, we said this morning that red blood cells are these discs that are a little bit caved in the middle. They don't completely go through, but they're caved in the middle. So they look like little donuts when you look at them under the microscope, right? Those are our red blood cells. All of these little things that you see here, these little chunks, those are all platelets. Those are involved in the clotting process. We haven't talked about that yet, but we will. Those are involved in the clotting process. You'll see those all over the place today. I was joking with my class this morning. The final is going to be just counting these. There you go. There's your red blood cells. Count them. I'm just kidding. Well, somebody else is standing behind you going 5, 25, 35, 137. Okay. So what we are going to do is we're going to do a differential white cell count. Works the best, and you're going to have the easiest time with this if you're in a good part on the slide. So even though we aren't making our own blood smears, the way that they work is that you put a drop of blood on a microscope slide, and then you take a second slide and you smear it across, right? So the idea is that at one end, the blood is going to be really heavy, and like blood cells are going to be stacked on top of each other. And then at the other end, they're going to be like too spread out, right? So we want to be somewhere in the middle to where the blood cells are roughly evenly spread out. Where I'm at right now is a pretty good space. Let me go over really quickly over here, all the way to the, whoops, wrong way. And this slide is actually pretty good in general. But if I go all the way over, notice here how stacked on top of each other those blood cells are. Some of them are like really stacked on top of each other. And then if I go all the way over to the other end, I keep going over how over here, like the cells are really spread out and they have big empty spaces? We kind of want to avoid that area too, right? So the best bet is to find somewhere in the middle where things are roughly evenly spread out. It doesn't really matter where you start on your slide. We want to try to find a place though where they're pretty evenly spread out. So let's go ahead and start right here. Trying to find a good starting point. Trying to find something good for you guys to start with. Ooh, there we go. Let's start here. All right. So this is a pretty good starting place. It doesn't matter where you start. It really doesn't. As long as you're in a place where they're roughly evenly spread out. So here I've got 1, 2, 3, 4, 5 white blood cells in this particular frame of view that I have with you. Okay? So let's go ahead and take a second and identify those. 3 of them are the same, right? So this one, and this one are all the exact same cell. So what I'm looking at here is I'm looking at a cell that is definitely, all of these cells are going to be bigger than red blood cells, but they're not overly large. But the nuclei on all of those are really unique, right? They look like, I tell you what I think they look like. Not judge me. They look like little cat tootsie rolls that are in there, right? Like little turds, right? That's kind of what they look like to me. They look like little cat turds. And I know that sounds weird, but that's how I envision them, and that's what I see, right? Is it's like what you might see when you're cleaning out the cat litter box. Is what you've got inside of the nucleus. So here we've got a nucleus that looks like there's a lobe there, probably a lobe there, and then maybe 1 or 2 lobes right there. Over here there's a lobe, and a lobe, and a lobe, and then over here at least a couple of lobes, if not more, right? They're definitely spread out. So which one of our white blood cells is going to have that really low nucleus? These are neutrophils, right? These are examples of neutrophils. Let me clear my little speckles so we can get a clean look at those. These are neutrophils. So what we're going to do as part of our differential white cell count is we're going to write down neutrophil. And in this frame of view, I have 1, 2, 3 neutrophils. Okay, and then the other 2 also appear to be the same. These guys are a little tricky, to be quite honest. I'm pretty sure these guys are going to be our lymphocytes. These guys are lymphocytes. They're not terribly small, but you'll notice that the nucleus in both of those takes up most of the cell. Okay, there's not a lot of cytoplasm that you can see around the nucleus, but more importantly, the nucleus is really round. Okay, so I think these are both lymphocytes. A little bit bigger than normal lymphocytes, but more round. So we have 2 lymphocytes. Okay, so there we go. So in this field of view, I was able to identify 5 white blood cells. So what I'm going to do is I'm just going to go up, start working my way up until I come across another white blood cell. It might be a minute. You know. Aha, there's 2 more. So I'm going to get those guys firmly into my frame of view. What are these guys? Those are your cat Tootsie Rolls. What do you think they are? Take a look at those guys. What are those guys? Really squiggly nuclei. They're multiple lobes. Those are neutrophils. Okay? So 2 more neutrophils. 1, 2 more neutrophils. And then I go up again. Okay. Oh, that's a beautiful, what is this guy? That is a lymphocyte. That one is like a textbook lymphocyte. couldn't have written that one better. There we go. So there's a lymphocyte. Again, big round nucleus, very little cytoplasm on both sides of it. And I go up a little bit more. Whoop. What are these guys? They're the same. Those are both neutrophils, right? So 2 more neutrophils. I'm trying to find us something good here. Okay, so the idea here, we'll go back to this in just a moment. The idea, you'll notice that what I've done here is I have my slide. I've started at a point and I'm working my way up. Okay? When I get up to the top of my slide, what I'm going to do is I'm going to go over one field of view, and then I'm going to start working my way down, one white blood cell at a time. And then I'm going to come over again, up, over, up, like this. What that prevents me from doing is just searching like willy-nilly all over the slide, and it prevents me from counting the same cells twice, right? So the idea is to do this until we get your lab book. Your lab book says 100. We're going to say 50. Okay. The idea is to do this until you count 50 white blood cells. Okay. 100 is a good number because it's easy to calculate percentages, but 50 is fair, I suppose. But the idea is to do this until you get to 50, right? So I'm going to come up, up, up. There's 2 more neutrophils. I think I wrote those ones down. What's this one? That's a neutrophil. With kind of 2, what looks like 2 nuclei. There's another lymphocyte. This is probably just junk on the slide. I don't know what that is. Anyway, I'm going to get, ooh, here we go. Haha. All right, what's this one? We know that one by now, right? That's a neutrophil. What do you guys think this one is? That is an eosinophil, yes. Notice how, and it might be hard for you to see, but there are 2 very distinct lobes to the nucleus, and then we have those distinct red granules in the cytoplasm. Okay, so that's an eosinophil. Okay, so we can add that to our list. I've gotten a little lazy with my counting. You can add an eosinophil. Okay. So then what I do is I get to the top, let's say for the sake of argument that I'm at the top of my slide, I come over and then I start to work my way down and look at all those lymphocytes. Holy shnikes. Batman. Holy lymphocytes, Batman. There's a neutrophil. There's another neutrophil, another neutrophil. 2 more neutrophils. A lymphocyte. Oh, there we go. Look at that bad boy. All right. So this is a lymphocyte, roundish nucleus. Look at this guy. Look at the nucleus here. C-shaped, and it's also really big. Who is this? That's a monocyte. Yeah, that's a monocyte. Okay. I'll leave him up so we can take a look at it. But that's a great example of a monocyte right there. Sometimes you will see the monocytes kind of stuck to red blood cells. Sometimes it actually looks like they're being pulled by the red blood cells. What they're probably doing is they were phagocytizing that red blood cell, and we kind of caught them in the act, right? So if they look like they're being pulled in multiple directions, it's because they were being caught in the act. But that's a great example of a monocyte. Okay. So differential white cell counts are how we look at white cell counts in blood. When you go give blood, somebody has to do this to your own blood to count your blood cells. Okay. With practice, it only takes a couple of minutes to get to 100, though. It doesn't take a lot of time. The goal for us today is to look at this one, and then after we're done with our normal blood smear, we'll look at the eosinophilia slide. And look at the difference between a normal blood smear and an eosinophilia. And the idea is that we'll see a lot more eosinophils in there. But it's also practice, right? Just learning to identify the differences between these blood cells. It takes time. So what I told my lab this morning is, and most people actually did this, that if you're still not feeling comfortable after you get to 50 white blood cells, go to 100, count 50 more, okay? If you're still not feeling comfortable after that, go to 150. They won't take you that long. Okay? It's not like you have any better. So, that wasn't very nice. It's not like you have a life. But anyway, but no, I mean, if you're not feeling comfortable, then do 50 more. It won't take you very long, and it's great practice to learn to identify. If you can't identify something, then just skip it and go on. Or ask me, I'll see if I can do it. Sometimes, though, they get broken and they become real messy and they become really hard. So, I don't worry too much about those. But the good ones are actually kind of fun to identify. So, in the back of the room, we have a couple of slides. We have a regular blood smear, and then we have the eosinophilia. The class this morning should have gotten everything back in the right tray. It's almost, not quite. Should have gotten everything back in the right tray. But just make sure before you start that you don't have an eosinophilia slide, because your count will be off. So start with the regular blood smear and then go to the eosinophilia next. Okay? So let's grab a scope. Let's do a differential white cell count.
I'm like, I never say thank you, but thank you.
Let's see what we
I
can
didn't